Hello and welcome to the inaugural talk in the Mathematical and Computational Ophthalmology seminar series. My name is Paul Roberts and I'm a Centre Fellow in the Centre for Systems Modelling and Quantitative Biomedicine at the University of Birmingham, where I work in the mathematical modelling of the eye. I'm the creator and host of this new seminar series, together with David Crabb, who heads the Crabb Lab at the City University of London, and Phil Luthert, a Professor of Pathology at the UCL Institute of Ophthalmology. Before introducing our speaker, Robert Linsenmeyer, I'd like to say a few words about the theme and aims of the seminar series. First, you may be wondering, what exactly are mathematical ophthalmology and computational ophthalmology? Well, computational ophthalmology is an existing term which involves the application of advanced computational techniques, including artificial intelligence, to the big data being generated in ophthalmology to identify pathology and predict disease progression. The emphasis here is upon prediction. Mathematical ophthalmology, by contrast, is a new term which I've coined and concerns the use of mechanistic mathematical models to derive insight into the mechanisms underpinning the structure and function of the eye in health, development, and disease. The emphasis here is upon understanding mechanisms. Thus, these two fields, mathematical ophthalmology and computational ophthalmology, provide complementary insights which are themselves complementary to those which can be derived through experimental and clinical studies. In both cases, the ultimate goal is to derive genuine translational impact in healthcare, accelerating progress towards more effective treatments, personalised treatment strategies, and clinical decision support systems. This seminar series aims to showcase research in mathematical and computational ophthalmology to the broader ophthalmological, vision science, medical, mathematical, and computational communities, raising awareness of the exciting research being conducted in the field and encouraging future work in the area. All past and future talks will be listed on our worldwide page, the link to which can be found in the About section of this YouTube channel. And our thanks go to Worldwide for instigating this platform for sharing scientific research. Last, if you'd like to ask Rob, our speaker, a question, then please do send these in via the YouTube chat, both during and after the talk. The talk will last for about 45 minutes, followed by 15 minutes for Q&A, in which I will relay your questions to Rob. So it gives me great pleasure to introduce our speaker, Robert Linsenmeyer, whose work provides a wonderful example of how mathematical modelling can be used to generate insights that would not be possible through experimental studies alone. So Rob is Professor Emeritus of Biomedical Engineering, Neurobiology and Ophthalmology at Northwestern University, where he continues to be active on many fronts. He's recognised for research in several areas of quantitative retinal physiology, particularly measurements and mathematical modelling of oxygen transport in the mammalian retina, for which he has been funded by NIH since 1983. He has published more than 130 refereed journal and conference papers and has a Google Scholar H-index of 44. Since about 2000, he has also devoted attention to engineering education research, particularly in the areas of undergraduate curriculum and design in biomedical engineering. He's received a number of awards for his work in education, including the Theo Pilkington Outstanding Educator Award and the Lifetime Faculty Mentor Award. He served in a number of important positions, including as the Chair of Biomedical Engineering at Northwestern and as the Founding Director of the Northwestern Centre for Engineering Education Research. He's also played leading roles in a number of national and international societies, including as Chair of the Biomedical Engineering Society National Meetings Committee and as Chair of the Academic Council, Chair of the College of Fellows, Treasurer and Vice President of the American Institute for Medical and Biological Engineering. He was also Editor-in-Chief of Current Eye Research and is an Associate Editor of Biomedical Engineering Education. So I'll hand over now to Rob to present his talk, which is entitled Unique Features of Oxygen Delivery to the Mammalian Retina. Rob.
Hi, everyone. Thanks very much for coming. And thanks very much to Paul for starting off this series and inviting me to give a talk. I really describe my work as mathematical and experimental ophthalmology, which both play a role in the things that we've tried to understand. So I'll share my slides, see if that works. All right. So as Paul said, it's called Unique Features of Oxygen Delivery to the Mammalian Retina. And Before I forget, I just want to make sure that I acknowledge people who have helped with this. So this is a partial list of the people who have been involved in this work. These are the people who contributed to the work I'll be talking about today. And the ones in purple are particularly involved, have been particularly involved in the modeling aspects of the work. As Paul mentioned, we've been funded by the National Eye Institute and in the early days by the Whitaker Foundation. So because we may have people who don't know much about the retina, I'm going to give you just a little bit of information about the retina. As most of you know, the retina is a layered structure with 3 cellular layers and 2 synaptic layers between them. And light comes in all the way to the back of the eye and is absorbed by the photoreceptors, which turn the signal into an electrical output that then propagates forward through the retina to the ganglion cells, whose axons form the optic nerve, and they leave the eye and go back to the brain. What's important for us today is the circulation. And the retina has 2 circulations. One is called the choroidal circulation. It's behind the retina. It's not within the retina. It's actually separated from the retinal neurons by the retinal pigment epithelium. And the other circulation is called the retinal circulation, which lies in the inner part of the retina, largely in the plexiform layers, that is the synaptic layers, although not entirely cleanly and exclusively. In humans, there's often a 3rd layer in the nerve fiber layer, but we don't tend to see that much in the animals we study. So it's convenient for us to think about the outer retina being the photoreceptors, which occupy about half of the retinal thickness of 225, 250 microns, and the inner retina, which is the rest of the retina, where the retinal circulation is. So if you keep track of outer retina, meaning photoreceptors, and inner retina, you'll find it a lot easier to understand the rest of the talk. So we're going to try to cover several topics today, answer several questions. Several of those are about the outer retina. The photoreceptors are in this layer that's 100 microns thick or more. And so the question is, where does our oxygen come from? Is it the retinal circulation or the choroidal circulation or both? And we're also interested in how oxygen utilization changes during illumination and what happens to the PO2 and metabolism under various disease conditions. And the one I'll talk about is when the retina is detached. Then we'll go on to talk a little bit about the inner retina, particularly oxygen utilization by the inner retina, and what happens in another clinical condition when there's a retinal stroke, that is a retinal artery occlusion. The first 2 questions will take a good bit of our time, so don't get nervous, we will get to the end. This is what the retinal circulation looks like when an ophthalmologist looks into the eye. Here the circulation is highlighted by fluorescein, showing the arteries lighted up on the left picture and the arteries and veins and capillaries all lighted up on the right picture. Most of our recordings that I'll tell you about are made outside the fovea. We know very little about what's going on with oxygen in the fovea. Most of our recordings are made out somewhat peripherally, where there's arteries, veins, and capillaries. In the fovea, of course, there is no retinal circulation and the retina is thinner. The retinal circulation has a flow rate and control mechanisms, much like cerebral circulation. Flow rates are 40 to 50 milliliters per 100 grams per minute. The metabolic control is dominant here. So when neural activity increases, flow rate goes up. That's called neurovascular coupling, which occurs in the retina as well as the brain. And the retina responds to high and low PO2s and high carbon dioxide levels as well, dilating or constricting the circulation. There's also good regulation in response to blood pressure and intraocular pressure, but no autonomic control. So that stops where the optic nerve enters the eye. The ophthalmic artery has a little bit of autonomic control, but circulation within the eye itself does not. The capillaries in the retinal circulation are normal, ordinary capillaries, but they have tight junctions between them, between the endothelial cells that makes the blood-retinal barrier. The choroidal circulation is completely different. The vessels for the choroidal circulation enter around the optic nerve, not through the optic nerve, the short and long posterior ciliary arteries. And microcirculation is also completely different. It's sort of a sheet of capillaries, more similar to what you would have in the lung than any other place in the body. These are large capillaries, they're fenestrated, and they allow a pretty high flow rate because the resistance is quite low. There is no metabolic control in the choroidal circulation. It does not care what the metabolic demands are of the retina, and its control is completely autonomic, largely sympathetic control. And the capillaries are fenestrated, so the blood-brain barrier at the outer part of the retina is at the retinal pigment epithelium, not in the choroid itself. So, okay, the first question, where do photoreceptors get their oxygen? So what we've done is to make measurements of oxygen gradients across the retina in vivo with microelectrodes. And then we've done mathematical modeling to obtain quantitative answers about how much oxygen is used and what its distribution is. And so this is the method slide to try to explain that in simple terms. We've done experiments in cats and monkeys and rats, relatively few monkeys, mostly cats, and in the last 10 or 12 years, and rats. These are all anesthetized animals, and they're paralyzed and artificially respirated. And we do that artificial respiration so that we can maintain the physiological parameters in the normal range by adjustments of the respirator. We start in dark adaptation always. And so we can't see the retina. We're not looking into the eye. And we rely on signals from the electroretinogram to tell us where we are in the tissue. The way we make measurements is with these double-barreled oxygen-sensitive microelectrodes. Nobody else in the world uses electrodes quite like this that we know of at the moment. One barrel of the electrode is a polarographic oxygen electrode that measures the current proportional to oxygen levels. It has very good spatial resolution for reasons I'm not going to get into. And the other barrel of the electrode is filled with just saline and is connected to voltage amplifiers to record the electroretinogram. So we put these electrodes through the wall of the eye around the pars plana, and then we can drive it into the retina, as I'll show you in a second. The top picture here is a cat eye, and the bottom picture is a rat eye, roughly to scale. It took us a while to figure out how to do this in rat, because the rats have beady little eyes, as you know. So here's a really critical picture to think about the way we do these experiments and what we get out of them. Here's our microelectrode in red at the bottom. What we've done is to put this into the retina in steps, checking the electroretinogram along the way. The electroretinogram changes with depth, so we know where we are in the tissue. And we can tell when we cross the retinal pigment epithelium also. So we use brief flashes of light for that, so the eye remains dark adapted between those flashes. And then we do a slow withdrawal at about 2 microns per second usually to generate the map of PO2 at the top. And we call that a profile of oxygen across the retina. The total thickness of the retina is 225 to 250 microns, but we often represent it in percentage retinal depth, with 0 being at the vitreal surface and 100% being at the choroid. So this profile is characterized by a few properties. There's a relatively high choroidal PO2 and a steep decrease in oxygen from that point down to the inner segments of the photoreceptors where all the mitochondria are, shown in light green here. There's a shallower gradient from the retinal circulation through this avascular layer to the same point. And in cats and monkeys, there's a very low minimum PO2 in the dark. In the inner retina, we have peaks of PO2, peaks and valleys, and the peaks correspond to being close to capillaries, but we can't see these capillaries. We just know they're there because these are relative maxima in the PO2 profile. So what do we do then with these profiles? We look at the outer retina. So here I'm focusing again just on the outer half of the retina. And you can see that that outer retina has a shape that's represented by the color picture down here at the right. So we start with just a diffusion equation that's one-dimensional, steady state, not remarkable in terms of its mathematical complexity. And the solution of that is that for each layer of a model, the PO2 is a quadratic term that involves the oxygen consumption of that layer and a couple of constants and distance. And the interpretation of the profiles is that if there's a straight line in the profile, as there is in regions 1 and 3, then consumption must be 0. Whereas if there's some consumption of oxygen, there will be a quadratic shape to the profile through that region. So with the profiles being linear in regions 1 and 3, we can drop the first term of the PO2 equation, and it simplifies to just a linear equation. But we need to solve the entire equation and use the second equation shown here for layer 2. So the full model has 3 layers for the outer retina. And the other thing we need to do is match the PO2 and the oxygen fluxes at the boundaries between layers. So what we do with that model then is to fit it to experimentally measured profiles of oxygen across the retina. And when we do this fitting, we are fitting 3 parameters, really. Well, there are 5 technically. The choroidal PO2, the PO2 at the inner border of the outer retina, the oxygen consumption in layer 2, and really the thickness of layer 2, the position of L1 and L2 between the layers. But the choroidal PO2 and the inner retinal PO2 are very tightly constrained by the data, so they're not really free parameters. And in essence, it's a 3-parameter fit that we're doing. Again, let me emphasize this is only for the outer retina. The inner retina has these peaks and valleys and is not really amenable to modeling, at least with this model. And the other thing to note is that the parameter values are actually fairly sensitive to the quality of the profile. We don't think they really change in the real world, but our profiles are sometimes not perfect. And so there is a fair amount of variability in Q2 and in the thickness of the of layer 2. But it turns out that if we get high values of Q2, that's always in a situation where the model is telling us that the thickness of layer 2 is relatively small. So Q2 and the thickness tend to be inversely related. And so we can reduce the coefficient of variation in the data that we report by multiplying those. So we tend to think about the oxygen consumption of the outer retina, which I'll call QOR for outer retina, as the product of the consumption in layer 2 and the thickness of layer 2. we divide that by L, then what we're essentially doing is saying this is the consumption that's sort of averaged over the entire outer retina, which is a useful number. Q2 is also a useful number, so sometimes we'll report that as well. So these are profiles from 3 different animals that we've worked on, macaque, cat, and rat. As you can see, the profiles are similar. Presumably, they would be similar in you as well. The average PO2 at the choroid is 47 millimeters of mercury in each of these animals, it turns out. And a key finding is that in darkness, 80 to 90% of the oxygen for the photoreceptors, and here we're talking about rods, comes from the choroid. About 10 to 20% comes down this shallower gradient from the retinal circulation. And this 80 to 90% and so on are results of the modeling as well. So because there's no good model for the inner retina in an intact retina, we tend to look just at the average values of PO2 in the inner retina. And this is what the distribution of PO2 values is for a bunch of profiles in cat retina. The average value is somewhere between 15 and 20 millimeters of mercury. So how does light affect this? Well, the PO2 around the inner segments, this layer shown in green, is higher in the light. And higher PO2 implies that less oxygen is being used. So oxygen consumption in the photoreceptors decreases during illumination. Sometimes in light, the choroid provides enough oxygen that it diffuses down this gradient that continues into the inner retina. But that's not always the case. What is always the case is that the PO2 in the inner segments increases in the light. The same diffusion model works, of course, and so we can extract values for oxygen consumption in both dark and light. And here are some important results of that modeling. The oxygen consumption of layer 2, that Q2, is about 20 milliliters per 100 grams per minute. That's a very high number. There's hardly any place in the body where you will find a number that's that high if you could figure out the local oxygen consumption. However, the weighted average for the whole outer retina, with much of the outer retina not using any oxygen at all, is only 3 to 4 milliliters per 100 grams per minute. And that's not a remarkable number. That's very similar to what cerebral oxygen consumption would be. Again, 80 or 90% of the oxygen comes from the choroid in the dark. And frequently, the choroid is able to supply a little bit of the inner retina in the light. When we look at the actual values of oxygen consumption in the light and dark, the value in light is 50 to 70% of the value in the dark. So it's considerably lower. And the other important part about the diffusion equation is that it provides the basis for the simulations that we do when we can't do experiments, or when we didn't think we could do experiments and later figured out how to do them. But before I get to that, let me spend a minute on explaining why metabolism decreases with light, which is a kind of an odd concept if you haven't thought about it before. The rods are relatively depolarized in darkness because they have a very high sodium conductance in their outer segment. So a lot of sodium flows into the photoreceptor in the dark, and the photoreceptor has to get rid of it. So the sodium-potassium pump that's in the inner segment is very active under dark adaptive conditions. The pumps are only in the inner segment, which is this layer where the mitochondria are, and the channels that allow sodium in are only in the outer segment. So there's this constant cycling of sodium around the photoreceptor. And when you turn on a light, what happens is that the channels close, less sodium comes in, and the pump, which is known in most tissues to be controlled by intracellular sodium, starts to slow down. So metabolism can also slow down when the pump rate slows down, because the pump is the major user of ATP in the photoreceptor. So what's the time course of this, and what's the dependence on the amount of light? In order to look at this, we've not measured profiles. Instead, we've left the electrode stationary at a point in the retina, out in layer 2, out near the inner segments or the mitochondria. And we've changed we've gone from darkness to light for 90 seconds or 2 minutes. And these are results of those experiments in monkey and in cat. So this is a single set of traces from a monkey and a single set from a cat. As you can see, when you make the light brighter, the PO2 goes up more in both animals. That means the consumption is going down more when you make the light brighter. The amount of light we use when we do a light adapted profile that I was talking about before are indicated by the stars here. And when you use that amount of light, there's a rather rapid recovery when you go from light back into darkness. If you use brighter light, then the PO2 does not recover quickly. Instead, it takes a very long time to come back down. The photoreceptors get saturated, and it takes them a long time to come out of saturation. So 10% of the maximum change, which is below the levels that I'm actually showing you here, corresponds to about 8.4% of the channels closing if you look into the literature on monkey photoreceptors. And that's far more than you need for rod vision. That's actually a fairly bright light. So within the normal range of rod vision, metabolism is really about the same as it is in darkness. So the little bits of light you need to see the moon, for instance, do not change your photoreceptor oxygen consumption. But during bright light, that is when you're using cones, the rods are saturated and they use much less energy. So let's go on and look at what happens to the metabolism and the PO2 when the retina is detached. Now, the point is here that the retina is not physically attached with any kind of junctions to the RPE. So it's easily detached from the back of the eye. And the detachments can be actually several millimeters wide. So with the photoreceptors relying so much on the choroidal circulation, what does this do to photoreceptor metabolism? It's also worth noting that detachments are not uncommon. They occur in diabetic retinopathy, in retinopathy of prematurity, and also in high myopia, if your eye is just a little too big. The way you deal with these is by doing attachment surgery to reattach the retina, but the vision may not return completely to normal, particularly if you have a macula off detachment. So what we've found from simulations is that even a small detachment will reduce photoreceptor metabolism. So the red curve here is a simulation of a dark adapted profile in the retina when it's attached. So the inner retina is not being shown here at all. This is just the profile through the outer retina. And we'll say that the oxygen consumption of the photoreceptors there is 100%. We can then detach it by 100 microns or 500 microns. And then the outer retina is shown by the solid line, and the fluid under the retina is going to have a gradient through it, because that's the way diffusion works. And that distance reduces the flux to the photoreceptors. And so if it's only detached by 100 microns, you can't maintain an oxygen consumption that's more than about 34% of normal. And when it's detached by 500 microns, you would be down to 15% of the photoreceptor normal oxygen consumption. And that's only because now the inner retina, which is assumed to ride along with the inner retinal vessels, which are assumed to ride along with the detached retina, will be able to take over a lot of the metabolism of the photoreceptors. So shallower gradient implies a smaller flux and not enough reaches the photoreceptors. But we think hyperoxia, breathing 100% oxygen, would help in this situation. It's been known from several studies in rats and monkeys and cats and even pigs that choroidal PO2 is not regulated. And when you breathe higher levels of oxygen, choroidal PO2 goes way up. And so we can simulate that the same way we simulate those air breathing profiles. And now with oxygen breathing, the choroidal PO2 is higher, the flux is greater, and so you can maintain a much higher level of oxygen consumption by the photoreceptors. So we didn't think originally we'd be able to measure this. We thought it would be too complicated, but then we figured out that we actually could measure it. So these are profiles from a cat where we've made a measurement in the attached retina, and then we detached the retina by injecting fluid under the retina so that the detachment's a couple hundred microns thick. And now there's a fluid layer under the retina. Choroidal PO2 is much lower than it was in the attached retina, but here the animal is still breathing air. Then if we give the animal oxygen to breathe, PO2 goes way up in the choroid, goes up through the whole fluid layer, and the choroidal PO2 is higher as well. Now in this particular case, our electrode must have been close to a relatively large vessel. And so the inner retinal PO2 here was also pretty high. But the main point is that the choroidal PO2 goes up, and so the PO2 in the outer retina is assisted by breathing 100% oxygen. These green lines are interesting to me, but maybe we'll come back to that. If you look at the consumption that you can measure from these profiles, and again call the normal oxygen consumption 100%, when the animal is breathing air, the detached retina has only about, has a much smaller oxygen consumption. But if you give it oxygen to breathe during the detachment, you can help the oxygen utilization of the retina quite a bit. It varies with the detachment height and for other reasons that we don't necessarily know, but hyperoxia always helps. We also know from other work that if you do a detachment in a cat and leave it under 70% oxygen for 3 days, that you preserve photoreceptors in that animal's retina. So we think that that hyperoxia would be a good thing to use during detachment. Photoreceptor metabolism is normally reduced during a detachment, but hyperoxia can maintain it much better. Whereas if you do no treatment, you're likely to have photoreceptor loss. So we think it would be a good idea to treat people with hyperoxia while they're waiting for retinal reattachment surgery. So I wanted to talk a little bit about oxygen utilization by the inner retina. As I showed you, there's peaks and valleys in the inner retina. So the one-dimensional model that we can use for the outer retina doesn't really work for the inner retina, although it has been tried, but we don't really like it. There are other ways to get at the inner retinal consumption, but there are questions about those as well. So we wanted to address this in a different way. On the left at the bottom is a map of PO2 in 2 dimensions, where you see these peaks and valleys in the inner retina. The left side of the graph is the back of the retina, the choroid. And there are these peaks that correspond to capillaries. But suppose we got rid of those capillaries, suppose we got rid of the retinal circulation, then we might have a situation where we could, again, use a one-dimensional model to look at oxygen across now the entire retina. So what would our thinking be about that? Well, we know that the inner retina is mainly supplied by these capillaries in the inner retina. And if we get rid of it, there would not be enough oxygen coming from the choroid to supply the inner retina. But if we have an animal breathe oxygen, then we might be able to get the PO2 high enough to drive oxygen all the way into the inner retina, just as we did in the situation of detachment. And before we started working on this, there was some previous work that showed that this might be feasible. So how did we do this? So here's a cat eye again. And what we did was to introduce another probe into the eye. This is just a glass probe with a little ball on the end that we could push on a branch artery at the optic nerve head and eliminate the circulation, eliminate the retinal circulation in part of the retina that we could then record from with our microelectrode. So before we did the occlusion, we could see these peaks and valleys in the inner retina. After we did the occlusion, or during the occlusion, the choroidal PO2 and the outer retina were not affected, but there was no oxygen at all in the inner retina until we started using oxygen for the animal to breathe. And then the choroidal PO2 goes way up as predicted, and oxygen diffuses much further toward the vitreous from the choroid. It's a little better in light adaptation because metabolism is lower in light adaptation. Although in this particular case, the choroidal PO2 was also higher. Now, does this completely save vision? The answer is probably not, but it does a better job than breathing air. So this is an index of the vision. This is the B wave of the electroretinogram generated by the inner retina. During air breathing, that's almost lost after detachment. Whereas if you breathe oxygen, you can get back some of it, or in some cases, all of it. So on average, about half of the function of the retina. So there's still problems during occlusion, but the retina is much better off than it would have been breathing air. And a critical thing for us now is that we've eliminated the retinal circulation. So now we can apply a model that's one-dimensional and extend it to have a fourth layer that is for the inner retina. And it turned out that one layer was enough to describe the inner retina. There was no advantage in going to 5 layers in the cat retina. And the interesting result here is that the inner retinal oxygen consumption, QIR, tended to be pretty much the same as the oxygen consumption of the outer retina in darkness. So you often hear that photoreceptors use an extraordinary amount of energy, but in fact, the inner retina is using as much oxygen as the outer retina is in the dark. The inner retina does not change its metabolism in steady light, whereas the outer retina reduces its metabolism in steady light. So then the last question is, what's the implication of this for a person with a retinal stroke who has lost their retinal circulation? Well, we know that there's no redundancy in the retinal circulation and no oxygen supply to the inner retina from the choroid during air breathing. So what you wind up with is a serious scotoma if you have a branch retinal artery. You're blind in a portion of your visual field, or you can have complete blindness in that eye if you have a central retinal artery occlusion. And people think there's nothing much you can do about this. So Hera said that currently we have no treatment with definitely proven beneficial effect. So people have tried different things, but nothing has been shown to work reliably. However, hyperoxia ought to help. The clinical experience with hyperoxia is fairly mixed, so it's not a routine treatment. And some studies that show benefit have used hyperbaric oxygen, which is not generally available to clinicians. So what can we learn from the simulations using the 4-layer model? Well, here's the situation again. If we give a person oxygen to breathe, we should be able to oxygenate the whole retina. And it's critical to think about the fact that the retina can survive an occlusion much longer than the brain. So Hera and others have shown that you can withstand a retinal artery occlusion for 100 minutes or more and still recover full function, which is way longer than the brain could stand having its circulation interrupted in stroke. Could you use hyperoxia indefinitely? Well, the answer is no, but the vessels will often recannulate and open up due to natural thrombolytic processes. So if you can keep the retina alive while that's happening, you may be able to get some benefit. Here's what the modeling shows. So here's a curve for the oxygen gradient after an occlusion when a person is breathing air. The inner retinal oxygen consumption is 0. If the person breathes 100% oxygen, based on the parameters from monkey and cat anyway, you'd be able to achieve an inner retinal oxygen consumption of something like 80% of normal. Now you could get to 100% of normal if you breathe oxygen at slightly elevated pressure, but again, that's not generally available. So we think that hyperoxia should be beneficial. If you're breathing oxygen through a nasal cannula, even if it's running at 5 liters per minute, that's not going to do it. That's likely to get your choroidal PO2 up to only a little over 100. And you can interpolate a curve here, which would not provide sufficient oxygen for the inner retina. So it has to be 100% oxygen that you're breathing. Now, this high oxygen level is only going to be present for a short time. And if you end hyperoxia, PO2s are going to go back down relatively quickly. So under that condition, suppose you try to oxygenate the vitreous. If you're breathing at 2 atmospheres of oxygen and get the PO2 in the choroid way up, then what you're going to find is spillover into the vitreous. And maybe you can oxygenate the vitreous well enough that after you turn off the oxygen, oxygen will diffuse back into the retina from the vitreous. So does that work? The answer is probably not. It takes a long time to saturate the vitreous. So here, I'm looking at distance from the retinal surface out into the vitreous. After an hour or 2 hours, you're gradually seeing the PO2 in the vitreous come back up. But it will also gradually come back down once you reduce the PO2 at the surface to 0 by going back to air breathing. And so these curves will slowly get oxygen into the retina, but too slowly for the metabolic demand of the retina. So we think that oxygen at 1 atmosphere should be beneficial, but it requires delivery of oxygen for an extended time period. And that has never been tested properly in the clinic. There are things to worry about, but it would be better than nothing. 100% oxygen is generally available. The time window that you have after the occlusion is much longer than what you would have in the brain. And with no treatment, your retina will not survive at all. I want to return to the intraretinal oxygen consumption one more time, because it's possible that intraretinal oxygen consumption that we measured in the cat during occlusion is not normal, even at 100% oxygen. Remember that we could not sustain the B wave at normal levels during occlusion, even with 100% oxygen. So suppose we eliminate the circulation and we oxygenate the retina from both sides, that is, from the vitreal side and the choroidal side. That's not possible in vivo, but if we isolate the retina, which we've done in the mouse, from the mouse, then we can provide oxygen from both surfaces of the retina and possibly sustain normal metabolism throughout the tissue. So the black lines here are single profiles recorded in mouse retina. It's the same profile on both panels of this figure. And it's fitted with the model that I'll show you in a second. It's a 4-layer or 5-layer model now, and the fits are very good. The perfusion fluid does not need to be hyperoxic. It can have 21% oxygen in it. And then we can see what happens when we try to fit these profiles, which have no retinal circulation, with a 4 or 5-layer model. It turns out that the fit is substantially better with a 5-layer model. This is an average set of profiles from dark and light that are reconstructed using the parameters that we have obtained from the fits. And the inner retina here is divided almost in half into a layer having Q4 and a layer having Q5. And interestingly, the consumption of the innermost layer, the one closest to the vitreous, is significantly less than Q4. And in many profiles, it's actually 0. So if we look at the intraretinal consumption, QIR, which is going to be a combination of Q4 and Q5 in the mouse, if we look at QIR from the cat retina, it's 3.5 milliliters per 100 grams per minute. And we have almost exactly the same number from the isolated mouse retina. So we think this is a pretty good number now for the intraretinal oxygen consumption. The outer retinal oxygen consumption from the mouse retina is lower than it is for cat. We don't know exactly why this is, but when you detach the retina from the retinal pigment epithelium, there's always going to be some photoreceptor damage. And so we think that's possibly why the mouse retina has a lower consumption than cat retina. The density of photoreceptors, the type of photoreceptors are the same in those two retinas. So it has to be something that we're doing to the retina experimentally that's responsible for this. So we're done. Retinal oxygenation is really pretty unique because of this dual circulation and because the photoreceptors have a metabolism that's altered by light. Fitting diffusion models to experimental measurements provides values for inner and outer retinal oxygen consumption and a lot of spatial detail that you can't get in any other way. So in darkness, the choroid supplies a lot of that oxygen. In light, it might supply all of the oxygen for the outer retina. Inner and outer retina have similar oxygen utilization needs. The outer retinal oxygen consumption decreases in light, which many other people have found as well. And what we found most recently, and I didn't have time to show you, is that inner retinal oxygen consumption increases in light, but only if the light is flickering, not if it's steady. It's also interesting that retinal oxygen consumption is similar to the oxygen consumption of the brain. It's not really extraordinary. Although photoreceptor inner segments, if you isolate just that little piece, do have an extraordinary oxygen consumption. And then oxygen measurements and modeling provide insight into retinal detachment, vascular occlusion, and several other diseases. So we think that there are good possibilities for therapeutic use of oxygen in retinal detachment and vascular occlusion, and now we just need to get clinicians to use oxygen intelligently. So with that, I will stop. Thanks very much.
Oh, well, thanks, Rob, for a wonderful and really clear talk there. So a number of questions have come in whilst you were speaking, and if anyone else has any further questions, please do type them in now. So I'll just take the first question here. So Donald asks, will slides be shared upon request? Would you be happy to do that, Rob?
Yeah, well, the talk is evidently going to be available on
yes,
on YouTube, so they're there. I'm not sure if you need the slides, but I could share them. Yes.
Thanks. Yeah. So and that actually comes back to another question from Ricardo. So yes, so the talks that now are being recorded, so that the recording will be maintained here on the YouTube channel at the same link. So you can, if you're not able to hear a talk live, then it'll be here indefinitely. So we've got a question here from Miranda Lynch. So she asks, the modelling helps us with understanding oxygen diffusion in these tissues, but can we couple this with reaction kinetics modelling for understanding the metabolic requirement or uptake of oxygen?
Yeah, so these, my oxygen consumption models are all zero order. So they assume oxygen consumption is constant down to 0 PO2. People have tried to use first order oxygen kinetics models. I don't think they get us very far, very much farther, because the half saturation of the kinetics is at such low PO2 that you really can't see much difference. Yeah, that's that might take a little more time to to go through if we were going to do it properly. But I think the reaction kinetics modeling doesn't help us very much.
Sure. Thanks, Rob. So we've got another question again from Miranda. This is so can we use the cat oxygen profiles for the detached retina case to validate the simulation modeling? How similar are the measured versus simulated curves?
Yeah, so they're quite similar. The simulated curves could be simulated with a variety of different choroidal PO2 values. We see that variety of choroidal PO2 values in the experimental measurements. So I suppose one could, and it also depends on distance. But in general, they're qualitatively close, and it's hard to do a quantitative comparison because it depends on the PO2 that one gets to. during hyperoxia. That's very similar to what the simulation is very similar to the experiments there. But the height of the detachment is very hard for us to control when we do experiments. So we only have a small range of detachment heights that we can actually do experimentally. I think the critical thing is the experiments that they did in cats with large detachments, where they showed that 70% oxygen, not even 100% oxygen, was really beneficial in keeping photoreceptors alive. So that in vivo sort of long-term study is a really key piece of why we think this is an important thing to try.
Thanks, Rob. So we've got a question from Ricardo who says, is the 2D model based on the assumption of axial symmetry?
All of the models I showed you were one-dimensional.
There was one perhaps with some lateral variation. Maybe he's referring to that.
Yes. So we do not allow any lateral variation. Our models assume that in the transverse direction to the two transverse directions to the x-axis, so in y and z, we assume that things are uniform and don't vary. So these are all exclusively one-dimensional models. Now we have done a two-dimensional model to look at what happens when you have a drusen under the retina and you have a hump there. The results of that are published in a review article that we wrote in Progress in Retinal Research. But mostly we stick to one-dimensional models.
Thanks. So we've got a question from Donald. So he asks, retinal oxygen species, it's not explained further, so I guess he's talking about the fact that you're using perhaps hyperoxia there and relating to that, I would guess.
Sorry, what's the question?
So the question is retinal. So the question is retinal oxygen species. So I think that's presumably that's relating to when you're using hyperoxia, and potentially.
Well, so we're only looking at O2. We're not looking at oxygen radicals or anything related to that. This is just molecular oxygen. That's what is being used by the mitochondria to generate ATP.
So Christian asks, thank you for an exciting seminar. He says some animals lack inner retinal vessels, for example, guinea pigs, birds and fish, and rely only on choroidal oxygen diffusion. Have you applied your approaches to some of those species to understand how oxygen can be supplied to the retina without inner retinal vessels?
Yeah, so that's a good question. We have not. We've been interested only in what are called holangiotic species with a retinal circulation, those being much more relevant to humans. The group in Australia, you and Kringle and those folks, have done work on guinea pig and rabbit, which also has no retinal circulation, except around the optic nerve head. And it turns out that those animals just don't use much oxygen in the inner retina. They are very heavily glycolytic in nature and have extremely low oxygen consumption in the inner retina because the oxygen from the choroid just does not make it into the inner retina. We don't know much about fish and birds in this respect. Birds, of course, have another potential supply from the pectin. But for guinea pigs and rabbits, their inner retina is heavily glycolytic and they have very low needs for oxygen in the whole inner retina.
Thanks. So I've got a question here from Jason. So he says, are there any scenarios where you envision that a central retinal artery occlusion would cause outer retinal toxicity or death due to hyperoxia? From your modelling, it seems not. He says there are rare clinical circumstances where it appears outer retinal thinning on OCT happens as a late consequence of a central retinal artery occlusion. Would that only be explainable by concurrent poor choroidal circulation?
That's interesting. So I did not know about that. One would not predict from the oxygen models that the outer retina would be particularly damaged, except that the photoreceptors do get 10 to 15%, 10 to 20% of their oxygen from the retinal circulation. So if you lose retinal circulation, you are potentially going to impact that 15 to 20% that the photoreceptors need from the retinal circulation. And if they can't make it up with glycolytic processes, that's going to hurt them. It turns out that in diabetes, a related concept here is that there tends to be damage to the deep layer of retinal capillaries. And there is also damage to photoreceptors there. And it may be due to the fact that the retinal circulation, the retinal supply to the photoreceptors is compromised under those conditions. So one possibility is that in the situation you're talking about is that the loss of retinal circulation impacts the photoreceptors directly. It's also possible that the inner retina is producing some reactive oxygen species or something that's toxic to the photoreceptors that then diffuses back and hurts the photoreceptors. So there are a couple of possibilities there, I think.
Thanks, Rob. And so I think we've got one last question. This is from Wei Li, and she says, great talk, thanks. She says, there are big mitochondria in photoreceptor terminals in monkeys and rats, etc. Why is oxygen consumption not detected in that region?
Yeah, okay, so this is a detail I glossed over. Those mitochondria in the outer plexiform layer are certainly prominent and very important. They are supplied by the retinal circulation, though. They are not supplied by the choroid, and they don't figure in our three-layer model. So they are in the, effectively, those terminals are in layer 4, effectively in the inner retina, because they're entirely supplied by the retinal circulation. So we don't see them in our three-layer model. Our layer 3 is the outer nuclear layer, which is, as you know, several stacked layers of cell bodies, and it doesn't take into account the synaptic layer of the photoreceptors, which we just include in layer 4 and not in the outer retinal model.
Thanks, Rob. So we'll finish the questioning there. So thanks again, Rob, for a wonderful talk and for your time in answering each of those questions. And before we close, I'd just like to advertise our next seminar. So this will be given by Aaron Lee on the topic of deep learning applications in ophthalmology. And this will be broadcast live at 5 p.m. GMT on Friday, the 10th of March. Please note that this is different to the previously advertised time, and web adverts are in the process of being updated. Further details of all future talks can be found on our worldwide page, the link to which can be found in the About section of this YouTube channel. And do also subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. So thanks again ever so much to Rob. Thank you to you for listening, and see you next time on mathematical and computational ophthalmology. Goodbye.